Good morning, everyone. Good morning. Welcome to this Coffee Time webinar, which is going to introduce the Bacterial Plant Diseases program. So my name is Geraint Parry, and I am part of the coordination team for this program. And I'm joined today by Sarah, Joanna, Laura, and Mariella, who will contribute to the talks a little later on. So we're all part of the coordination team as well. They'll be waving. So basically what's going to happen, I'm going to go through and give a very brief introduction to the program and also to each of the 9 projects. And then after that, the other members will come back and hopefully we'll take some questions from everyone at the end as well. So I'm going to say goodbye to everyone else now and then continue, and we'll see them all a little later on. So thanks, guys. Okay, so the Bacterial Plant Diseases program is a program which is funded by BBSRC, by NERC, by DEFRA, and also the Scottish government. And from the breadth of the interests of those sponsors, you can see that this is not just looking at the science of bacterial plant diseases. It's very much looking at both the nuts and bolts science, the interaction with the environment, and also interactions with stakeholders as well. So it's a very broad-based projects. We're really cognizant of how environmental changes will change the challenges for plant health over the coming years. So therefore, this program, which is sponsored through the Strategic Priorities Fund, has been put together to basically prepare and do research in this area. So what I'm going to do this morning is just to go through very briefly each of the 9 projects, just to kind of whet the appetite for everyone to the projects that have been funded. And then, we'll hopefully inspire you to get in contact with either the coordination team or with the project PIs to see where you might be able to get involved with these things. So all of these slides are available for download from the GoToWebinar software, if you'd like to do that. And also, we really like this to be interactive. So at the end of the presentation, which should take about 25 minutes, we're going to take questions. So please type any questions you have into the question box. If we have a deluge, hopefully we will, but if we do have a deluge of questions, then we will be sure, if we can't get to all of them, then we'll be sure to reply to everyone online afterwards. Okay, so there we go. So the Bacterial Plant Diseases projects, they have been funded in 2 phases. So the first phase was funded a couple of years ago, and that was the Bridget project that you may be familiar with. So that's been active for a couple of years and will finish next year, and is really the largest consortium of all these projects, 12 different organizations involved in it. The phase 2 projects have only just been funded, and they began basically at the start of the summer, some of them are beginning around now, and will run for 3 years. And they're really, as I stated, the idea of this is they're really multidisciplinary projects. So multiple academic institutions, government, SMEs, and larger industry as well involved in each of these projects. So as I said, I'm going to go through each of these projects in detail, in a small amount of detail, just to kind of whet the appetite, I guess. So the first project, this phase 1 project, which has been ongoing for a couple of years, is the Bridget project, and this is involved with surveillance and response to Xylella. So Xylella is a broad host range bacterial pathogen, which is invasive in Europe and is transmitted by sapping insects like leafhoppers or spittle bugs. So it's been a real problem in the Mediterranean regions, kind of devastating the olive groves in Italy, and is now moving through Europe and predicted to reach the UK pretty soon. So this program was put in place. This project was put in place in order to really prepare ourselves for the possibility of this bacteria reaching our shores, basically. And there's really 2 main phases to the project. So really looking at the biology and the phylogenetics of either the bacteria, the Xylella itself, or also for the sapping insects as well, so the insect vectors. So really 2 main portions of the project, which obviously come together, which will allow us to plan kind of management strategies for this. So as I said, this project has been going on for a couple of years now. It's led by Saskia Hugenhout at the John Innes Center, and with a broad set of collaborators. But they have a website, and there's a lot of information on there already, because as I said, this has been going on for a couple of years. And we're really hoping that the Bridget project can be a real exemplar for what we hope can happen for the other bacterial plant diseases projects, in that they've been really excellent in sharing the protocols that they have, in also sharing and setting up some information about genomic resources and things. So hopefully the other projects can follow along with this, in this kind of sense of idea of openness, really. Okay, so that was the phase 1 project. So really the highlighted outcomes that we'd like to see from this are to increase awareness, the identification and detection of tools for Xylella, generate information about insect vectors, and then ultimately develop models to inform surveillance and then control strategies. Okay, so for the 8 phase 2 projects, the first one I'd like to discuss, in no particular order, they're just the way they kind of link together, is the Calibrate Project, which is led by Adrian Fox at Ferra Science. And this is also involved with an insect vectored bacteria. And the focus on this, so the bacteria is Candidatus liberabacter, and the main host for this bacteria is, and the main experimental host, at least for this project, will be for carrot. So there's an insect vector involved, but we have very little information about that. So this project will be again looking at the strains of the bacteria, also looking at the insect vector, and putting these two things together in order to come up with a plan to tackle this. So the aims and outcomes of this project will be to improve the diagnostics, the detection, improve the knowledge basically about the incidence and epidemiology, since we don't know a huge amount of this at the moment, and then ultimately model how we might be able to deal with the threat from this disease in the future. So the predicted outcome, the highlighted outcome, will be to have tools for predicting outcomes and primary surveillance for this bacterial infection. So the next project is the Backstart Project, and this is related in that there's also an insect aspect to this. So there's a beetle that vectors this bacterial disease, and the bacterial disease in this case is acute oak decline. This project is led by Sandra Denham, who works at Forest Research, and this project looks really across the gamut for those areas involved with acute oak decline. So there'll be first of all in work package 1 looking at the interaction between the beetle vector for the bacteria, also looking at the bacteria and the looking in detail at that pathosystem, but also then looking more broadly. Obviously, oak is the major deciduous tree in the UK, so any management practice that comes in to prevent acute oak decline will have significant impact on those oak forests and people who deal with that aspect. So they'll be looking at the management decisions, and also be looking a little more broadly about the effect of acute oak decline on other tree species as well. So the outcome will be to develop management advice on beetle control and managing the stress in trees. So very much linked to the Backstart program is the Future Oak program. So this is also looking at acute oak decline, and this project is led from Bangor University by James McDonald, and it does include many of the same collaborators, many of the same partners that are involved in the Backstart project. Although in this case, this project has a slightly different focus, and very much they are looking at developing this idea of suppressive soils or suppressive microbiome. So the idea that the microbial populations that interact with both the tree and interact with the soil can have a protective effect on for the tree in the pathogens that it will interact with. So they're going to really investigate this in more detail, look at the microbiome, look at the bacterial populations that are there, look at the metabolome of these bacterial populations, trying to understand this system, and then ultimately try and look at again the management of this if this can be used as an effective control. So really, this kind of really novel project to develop suppressive microbial mixes that can protect these oak trees. So this idea of suppressive soils and suppressive microbial populations, beneficial microbial populations, are really the focus of the next project, which is led from the University of Sheffield by Duncan Cameron. And this project is also looking at this idea in detail about a suppressive microbiome. So how is it that bacteria that interact with the plant or with the soil can cause resistance to different pathogens? And they're really focusing on crops and also so in tomato as well. That's their main system. But this project both has the microbiome aspect, but also has this aspect of synthetic soil. So they already began to develop this synthetic kind of foam-based soil system that can be used as an alternative to conventional growth. And really those two strands are going to go along side by side in the aim of producing a novel growing system, whereas the beneficial aspects of the microbiome can be put together with this synthetic soil in order to develop, as I said, this new system. So initially they'll be characterizing the microbiome, understanding which bacteria are important, what the metabolome of these bacteria are important, and how then this can be fed into the development of this synthetic soils. So this project was launched in May, and it had a lot of interest in the mainstream media actually at the start. So this idea of plant probiotics kind of grabbed the attention. So hopefully this will continue throughout the project as it develops. So ultimately, the highlighted outcome will be to develop disease suppressive so-called probiotics for this bacteria. So the next project is also involves the soil. It's a kind of tenuous link between the two, but this involves the soil, but is led from the James Hutton Institute in Scotland by Ian Toft. And this is looking to develop a decision support tool for blackleg disease. So blackleg disease is so Pectobacterium atrospecticum is a disease of potatoes, and it has a significant cost to the UK agricultural economy. And the primary work that a lot of this project will be based upon is the fact that they've shown a link between blackleg and also between the levels of irrigation and the interaction with free-living nematodes. I guess the immigration of the free-living nematodes maybe, but so the interaction of irrigation and these nematodes in transmission of the disease. So this Blackleg project is maybe the largest consortium of the phase 2 projects, and really they're looking at many different aspects of this disease. So there's an insect issue is also looking at that, looking at these nematodes, looking at the effect of cover crops and also of the microbiome on the transmission of this disease. And ultimately everything will be moving forward into develop this decision support tool for farmers whose crops are in danger from this disease. That's really, so the whole project, even though there's a large number of collaborators all looking to develop this tool, this decision support tool. So ultimately that is the outcome. And they're recruiting a postdoc at the moment. So if you go to the Bacterial Plant Diseases.uk website, you can find a link there through their page. They're looking for a postdoc who's involved in light sheet microscopy, actually, so looking at that interaction. So that kind of highlights the real multidisciplinary aspect of this project. They're not only interacting with farmers, but they have some hardcore microscopy going on as well. So it's super interesting in that regard. So the next project, again towards the end, is the 3 more projects to describe. So the xanthomonas plant diseases project. So this project is led from the University of Warwick by Professor Murray Grant, and really they're taking advantage of the UK Vegetable Gene Bank, which is held at the Wellesbourne campus of the University of Warwick, where they have 14,000 different accessions of UK vegetable crops, which includes 6,000 different brassica accessions. And they'll be looking to test that germplasm on their susceptibility, resistance to xanthomonas infection. And then alongside that, there'll be a large-scale model sequencing project, which will go on at Exeter University. So sequencing xanthomonas accessions, and obviously putting those two things together to develop ideas about developing resistant germplasm and diagnostic tools for xanthomonas infection. So they're going to go further as well, not just focus on brassicas, but look. Xanthomonas has a broad host range, so look at UK maize and also into strawberry and other monocots on strawberry as well. So really looking more broadly about the effects of xanthomonas. So the outcomes for this will be to improve the molecular diagnostics ultimately learn more about this system so that this can inform policy and outbreaks in the end. Okay, so moving on to the next project. So the penultimate project also is involving a pathogen which has a real broad host range. And this project is led by Richard Harrison at NIAB. And this project focuses on the Pseudomonas prunus system. So the infection of cherry trees by Pseudomonas pathogens. So quite a bit is known about the life cycle, I think, of the interaction between Pseudomonas and prunus, but less is known about the real specifics of this interaction and how it's affected by the environment. So this project will very much focus specifically in this system, maybe a bit more specific than some of the other projects. So looking at these bacterial populations over time, what is it that influences their colonization of the cherry trees, nutrition, inoculation rights, different environments, what contributes to the host range of this, and then ultimately how can this information be extended to help us understand other Pseudomonas infections. So as I said, it's got a large host range, so how can this be extended to influence other systems as well. So ultimately the project will develop tools for the risk management and precision diagnostics of Pseudomonas infections. Okay, so the final phase two project is from the University of York and is led by Villa Freeman and Andrea Harper. And they're using a really cool technique to use phages as a precision tool to control pathogen abundance in plants. And in this system, they're targeting Rustonia species, Rustonia which infects tomato plants. And they've got previous research that shows that these phages are able to prevent infection of tomato by this Rustonia. So they're really looking to develop this system as part of this project. So they're going to extend this analysis, obviously understand how it works specifically, make sure that the phages do not have any effect on beneficial bacteria, really look at the evolutionary relationship between the phage and the bacteria to see if the phage can induce a less virulent form of the bacteria to grow. And really, not only were they looking at the biology, but they're also looking to extend this. So if this phage can be used as a biocontrol, obviously there's a lot of issues surrounding the manufacture, making sure that this is a safe thing to use. So they're really looking not only at the specific nuts and bolts of the interaction, but also looking much further downstream about manufacture and validation of this as a potential biocontrol. Okay, so that's a really quick run through the 9 different bacterial plant diseases projects, and I hope you found something interesting in there. So you're able now to, as I said, you can download this information. But now I'm going to pass on to Laura and Mariella, who are going to discuss about the impact and the stakeholder engagement aspects. Oh, there we go.
Great, thanks, Geraint. So I'm Mariella, and so as you can see, the Bacterial Plant Diseases Programme hosts a range of innovative and vitally important research projects. And as part of the coordination team, the role of myself and Laura Marr is to encourage strong communication and engagement between all those who are involved in, interested in, or could be impacted by the research. Most of the project started in September, and we have a wonderful opportunity here to work together across research, policy and industry to make sure that the outputs and outcomes of the research projects have real impact. And the aim is to encourage interdisciplinary and co-design approaches to research development to support the projects in their stakeholder engagement, as well as provide a platform for stakeholders to engage with all the different projects, just as we're trying to do here. So next slide, please, Geraint. Getting there. So you have a few activities to start with, including developing a stakeholder map, as social scientists always do, because we want to make sure that we're aware of, include all relevant individuals and organisations. Each project has its own stakeholder engagement plan, which is really helpful. And soon we're going to be sending out a very short survey to as many stakeholders as possible to get a sense of their plant health concerns and their information needs. So if you do receive a survey, we'd be grateful if you could fit in, because it will help us to understand how we can engage with you better. The stakeholder mapping exercise is going to feed into a document, iterative document if you like, where we're going to map the synergy and the overlaps between stakeholder interest and different projects, so we can look for opportunities to have joint events and engagement activities that work really closely with you. And the coordination team are also looking for opportunities to give this type of presentation or introduction at industry events, so please do get in touch if you're interested. And I'll say a little bit about that a bit later. So I'm going to pass over to Laura.
Okay, it's good to be talking with you. So you've heard about all the practical outcomes that the projects are seeking. And one thing we all share is that we want all the research projects to make a difference, to have an impact in the real world. And fortunately, we know a little bit about the fact that impacts come about through processes. They don't just suddenly happen. And because we know a little bit about these processes and how they usually involve interactions and collaborations with stakeholders, we're going to be on hand to work with the projects to help them think through how they can reach out to the real world and how they can understand and improve their processes leading towards impacts in the real world. So we're going to be asking everybody, the researchers and the stakeholders in each project team, we'll ask them some challenging questions about steps towards progress and towards impact and lessons learned. But even though we'll be challenging, we're always going to be constructive and helpful. So we're going to be the critical friends helping the projects, researchers and stakeholders together, lead towards impacts. And now Sarah Green, who's the leader of us all in the coordination team, is going to take over.
Okay. Hello, everybody, and I hope you can hear me. So I'm just going to really finish off by introducing again the coordination team and the rationale behind why there is a coordination team around these programs. So I'm leading the coordination team. My name is Sarah Green, and I'm a forest pathologist with Forest Research. And my role is on the sort of direction and delivery of the coordination team activities. I'm really a primary point of contact for the program management group, but I'm also really keen to engage with stakeholders to look to see how well the projects can align with what the stakeholders' needs are and what tools and management options can be improved by the end of the project. Murray Grant, who's not able to join us today, is the Elizabeth Creek Chair in Food Security at Warwick University, and Murray's role is to lead on the engagement with the science teams and the grant holders, and he's really keen to look at sharing of information, sharing of liaising, getting the science teams to liaise with each other and discuss the different methods that you're using to ensure that there's sort of sharing of information, sharing of resources where possible, looking at technology applications and data management as well. Joanna Vincent is also on our coordination team and is working with Murray Grant on engaging with the science teams. Joanna is a senior plant bacteriologist at FERA. So in addition to working with the grant holders, Joanna is also leading on the communication and publicity side of things. So Joanna, together with Geraint, have been putting together the website, and they're leading on the Twitter as well, sort of publicity. And you've met Mariella and Laura, both social scientists, and are very much involved in the stakeholder engagement and impacts. And I'd also like to introduce Debbie Fredrickson-Matika, who is a plant pathologist working with me at Forest Research, and Debbie's providing admin support, essential admin support for our coordination team. And last but not least, because you've been hearing him, is Geraint Parry, who is also very much involved in coordination of events over the next 3 years as part of the programme, and looking at organising meetings. He's also interested in developing training opportunities within bacterial science, and also involved in the website. So collectively, our role is to really ensure that this is not a series of 9 separate projects all sort of going their own way, but that actually everyone involved in this research considers themselves part of a program, part of a greater community of researchers into bacterial plant diseases, and everyone can sort of help each other and get to know each other, and you know, that should help the furtherment of the science as well. And as well as ensuring that there's looking at synergies across the science, we are also very much ensuring that the research is relevant to the different sectors. Those people who are involved in vegetable production, tree managers, for example, fruit crop managers, that the research will help them to manage their bacterial plant diseases. So I'll finish there and I'll pass on back to Geraint. Thank you.
Okay, thanks, Sarah. So yeah, as Sarah mentioned, we're really looking to interact with people as much as possible, and that includes right now. So if anyone has any questions that they'd like to ask any of us here, we have experts in impact and about the science as well, so that'd be really useful. But let me ask, so Sarah, can you come back onto the screen, and I can, there is one question that we have for this. So hey, so this is kind of an unusual program, the fact that there's these linked projects together. So what would you consider to be a success at the end of the 3 years? What would you like to see as a success at the end of these projects?
Yeah, I mean, that is a good question, is what does success look like at the end of a 3-year research program that's involved a huge amount of money going into it. And I think ultimately there's a number of indicators of success that we'll be looking for. One is clearly that we will have strengthened the science base in bacterial plant diseases, and that strengthening of the science base enables a much greater understanding of how bacterial plant diseases act and how we can manage them better. It will also help us hopefully to manage the existing bacterial diseases we have in this country. But I think looking at Xylella and looking at what we're dealing with for example, in forestry, it's this threat of emerging diseases that no one had ever heard of before until they actually arrived in the country. And that's the biggest, in some ways, the biggest concern. So I think building the science base will enable a much greater preparedness for these new and unknown bacterial plant disease threats. Other outcomes that we'll be looking at is that we have a cohort of younger scientists that have been trained up in bacterial plant pathology. That this is the future for the strengthening our future ability to deal with bacterial plant diseases in the next 10, 20 years and beyond. But ultimately also, every single one of the projects has some form of practical outcome that they are looking to, whether they achieve that within 3 years or they lay the groundwork to having a practical outcome available beyond the 3 years of the program. But ultimately we are looking towards working to practical tools to help in bacterial disease management, and whether that's looking at novel biocontrol methods, or for example disease phages or disease suppressive soils, whether you're looking at how to enhance an understanding of resistant cultivars, resistant to bacterial plant diseases, or essentially understanding how you can forecast diseases much better, and precision diagnostics as well. So I think ultimately we are looking at these outcomes at the end of 3 years, and we can say, yeah, this program's been successful. And everyone's talking to each other as well, still at the end of 3 years.
So yeah, I mean, hopefully this can be the way the coordination team can be kind of an exemplar also for future ideas. So larger programs, then maybe you can have a group that can oversee it in some way to identify synergies and things. So it seems to me that we've blown everyone away, and that no one's got any questions to ask.